In this video, we will discuss about effect of Cadherins uh, on the ECM depositions around the pancreatic cancer cells. So from the heading, it is clear that it's the Cadherin-3 that alters the ECM deposition in pancreatic cancer, and the major compositions of the ECM is collagen-1. So that's why the researchers focuses on the impact of this Cadherin-1 and Cadherin-3 on this collagen-1 around the pancreatic cancer cells. So first, what they did is they check the effect of these Cadherins on the tumor growth and tumor progressions. So they implant these knockout cell lines and the wild type control into the mice model, and then measure the tumor volume and tumor weight after different time intervals. And they are able to see that when Cadherin-3 has been knocked down, they are able to see a decrease in the tumor volume compared to the case where the Cadherin-1 has been knocked down, where the difference in the tumor volume between the wild type and the uh, Cadherin-1 knockdown is non-significant. And similar things they reflected in the tumor weight, where they can see that when the Cadherin uh, 1 has been knocked down, in that case they are able to see when the Cadherin-1 has been knocked down, in that case they are able to see the high tumor growth, whereas when the Cadherin-3 has been knocked down, the tumor growth is suppressed. And when they check the immunostaining in the to find out the impact on the ECM, they are able to see that the storma formations where the both Cadherin-1 and Cadherin-3 are present is high, and where only Cadherin-3 present is also high. But when only the Cadherin-1 is present, they see a low storma formations. And when they also check it on the primary tissues, that is CRCM 1110 that contains both Cadherin-1 and 3, and CRM08 that contains only Cadherin-3. And in both cases, they are able to see the ECM formations are high. So only conditions where ECM formations are low, where Cadherin-3 has been knocked down. And when they quantify these things, they are able to see that they are able to see the storma area formations is lowest. Storma area formations is lowest when this Cadherin this Cadherin 1 is absent, and similar things they have observed for this for the primary tissues, that is when this Cadherin 1 is absent, they are able to see a decreased amount of stroma formation compared to the case where both are present. So previously I have said that both of them are high, but when we compare CRCM 1110 and CRCM 08, we are able to see that the CRCM 08 has low storma because one of the Cadherin is absent. And in the this in subcutaneous injections of the tumors in the mice models, also we are able to see when the Cadherin 1 is absent, we are able to see less stroma formations. So when it comes to the tumor weight or tumor progressions, it's the Cadherin-1 knockdown that cause decrease in the tumor progressions or tumor weight. But when it comes to the ECM, we are able to see that when Cadherin-1 knockdown will cause less ECM storma formations. And in order to validate these things, they check the pancreas weight in different conditions. In the healthy conditions, the pancreas weight is considered as this 150, but when the case where both the Cadherins are present, that is when the mice is injected with wild type BXPC3 cell lines, they are able to see increase in the pancreas. And when Cadherin-1 knockdown is used, they are able to see further increase in the pancreas. And this is in line with the results that are on the left hand side of the panel A, that when the Cadherin-1 is knocked down, they see a high increase in the tumor weight, and when the Cadherin-3 has been knocked down, they see a decrease in the pancreas. And when they check the storma areas in these cell lines, they are able to see that storma formations, this is the immunostaining of the panel B, and the quantifications are on the panel C, where we can see that the storma formations is lowest when the Cadherin-1 has been knocked down. So together all this indicates that the Cadherin-1 plays an important role towards tumor progressions, and at the same time Cadherin-1 also plays a Cadherin this Cadherin-3 plays an important role towards this tumor growth or tumor progressions. That is, when the Cadherin-3 is absent, we see a less tumor growth and tumor progressions. Whereas the Cadherin-1 plays an important role towards these ECM depositions. So this heading is not completely correct. We need to also specify about the Cadherin-1 that Cadherin-3 plays an important role towards tumor growth and progression, and Cadherin-1 impacts the ECM or stroma formations around the pancreas. That's all for this video. Thank you for your time.